Ribonucleic acid is another important nucleic acid found in the cell. It occurs mostly in the cytoplasm. Mostly in the cytoplasm of the cell. Only a small amount is found in the nucleus. And in the nucleus, RNA is found specifically in the structure called nucleolus. Another important feature of RNA is that except for some viruses, RNA does not function as genetic material. We have seen that DNA is the genetic material in all living organisms, from bacteria to human beings. Nowhere RNA functions as the genetic material except in the case of some viruses. Then what is the function of RNA? RNA is involved in protein synthesis. Protein synthesis under the direction from DNA. We are going to discuss this in detail later, and this process is described, the entire mechanism is described as gene expression. RNA has a very important role in gene expression under the direction of DNA. RNA, unlike DNA, has only one polynucleotide chain. Only one polynucleotide chain. In DNA, we have seen there are two polynucleotide chains coiled around each other, but in RNA, there is only one polynucleotide chain. In RNA, the sugar is a typical pentose sugar called ribose, C5H10O5. So it is a typical pentose sugar, ribose, hence the name ribonucleic acid. The nitrogen bases in the RNA molecule are represented by adenine, guanine, cytosine, and uracil. Adenine, guanine, and cytosine are found in DNA also. In place of thymine in the RNA, you find uracil. That means RNA is composed of nucleotides of adenine, guanine, cytosine and uracil. RNA does not undergo replication. It cannot synthesize its own identical molecule, cannot undergo replication, hence it is to be synthesized only from DNA. These are the most important features of RNA. You can see almost in every feature there is a difference between DNA and RNA. DNA occurs mostly in the nucleus, RNA occurs mostly in the cytoplasm. Small amount of DNA occurs in the cytoplasm, small amount of DNA occurs in the nucleus. DNA is the genetic material, RNA is not the genetic material in living organisms. Only in some viruses it functions as the genetic material. Therefore, RNA has a very important role in another function, that is called protein synthesis or gene expression. In the DNA molecule, we have seen there are two polynucleotide chains that are coiled around each other, but in the RNA molecule there is only one polynucleotide chain. The sugar in the DNA molecule is deoxyribose C5H10O4. Sugar in RNA is ribose. It is a typical pentose sugar C5H10O5. Nitrogen bases. In DNA, nitrogen bases are adenine, guanine, cytosine and thymine. In RNA, nitrogen bases are adenine, guanine, cytosine and uracil. DNA undergoes replication, but RNA cannot undergo replication. Therefore, RNA will have to be synthesized only from DNA. There are three types of RNA. Three types of RNA. One is called messenger RNA. Messenger RNA, commonly abbreviated as mRNA. Messenger RNA forms about 5 to 10% of the total RNA in the cell. About 5 to 10% of the total RNA in the cell. It has about 300 to 5,000 nucleotides. Number of nucleotides ranges from about 300 to about 5,000. The sequence of nucleotides these nucleotides in the RNA will be complementary. To the code present in DNA. For each of the protein to be synthesized in the cell, DNA has a code. In this code, there is a particular sequence of nitrogen bases. mRNA is synthesized as a complementary molecule to this code. And in this sequence, suppose this is mRNA with a particular sequence of nucleotides containing adenine, guanine, cytosine and uracil. In this sequence, every 3 successive nucleotides represent what is known as a codon. Each of these codon specifies a particular amino acid to be incorporated in the protein molecule. When we go to the details of gene expression, we will know more about this. So this is the situation in the messenger RNA molecule. The second type of RNA is called transfer RNA. Transfer RNA, commonly abbreviated as tRNA. Transfer RNA accounts for about 15% of the total RNA in a cell. About 15% of the total RNA in a cell. It is the shortest form of RNA, containing hardly 75 to 80 nucleotides. Transfer RNA is involved in identifying picking up and transporting amino acids. Identifies the amino acid, picks up the amino acid and transports the amino acids to the site of protein synthesis. That is ribosomes. This property of tRNA molecule is very specific. The identification of the amino acid by the tRNA molecule is based on identification of the amino acid is based on the presence of what is known as anticodon. Remember, in the mRNA we have talked about codon. a combination of 3 successive nitrogen bases that specifies an amino acid. An anticodon will be complementary to the codon present in the mRNA. That means this anticodon is a combination of 3 successive nitrogen bases complementary to a codon of mRNA. Complementary to a codon present in the mRNA. The tRNA molecule is bent on itself. Bent on itself like a hairpin. In the year 1968, a biologist by name Hawley has given the structure of tRNA molecule to be in the form of a loop, this is the structure given by Hawley for the tRNA molecule. It is bent on itself. Therefore, at some of these regions, the nitrogen bases pair with each other according to the base pairing rule, pair with each other, and there are regions where unpaired nitrogen bases are present, forming loops. This loop is called DHU loop. DHU loop is rich in dihydroxyuridine. More of uracil is present there. Diagonally opposite to the DHU loop is a T loop. If there are about 12 unpaired nitrogen bases here, there are about 7 unpaired nitrogen bases in the T loop. Between the DHU loop and the T loop is the Anticodon loop. Anticodon loop. And in this anticodon loop, you find 3 nitrogen bases in a sequence forming what is known as an anticodon. This is the anticodon. Suppose this is the 5' end and the 3' end. The 3' end has a tail ending in CCA, cytosine, adenine, CCA, and this is the amino acid binding site. This is the amino acid binding site. This is the anticodon site. So based on the anticodon present, a particular amino acid binds at this region, and tRNA transports that amino acid to the site of protein synthesis. Therefore, the number of tRNA molecules will be equal to the number of codons involved in protein synthesis, because for each codon there must be an anticodon. Again, we will see that in detail later. The third type of tRNA molecule is called ribosomal RNA. Ribosomal RNA, commonly abbreviated as rRNA. Ribosomal RNA accounts for about 80% nearly of the total RNA. 75 to 80% of the total RNA content in the cell. It may have about 1000 to 1200 nucleotides. Ribosomal RNA is always found associated with ribosomes, and hence the name. So always found associated with ribosomes. So wherever ribosomes are found in the cell, you find ribosomal RNA. Ribosomes are the protein factories where amino acids are assembled in a sequence decided by code in the DNA. That message is copied by mRNA to form codons. With the help of anticodon, tRNA identifies and picks up the amino acids, brings them to the ribosomes, and ribosomal RNA helps in assembling the amino acids into a protein chain. So its function is to assemble the amino acids into a polypeptide chain. So these are the three types of RNA: messenger RNA, transfer RNA, and ribosomal RNA. All these three types of RNA are together involved in a function called gene expression. Gene expression is responsible for protein synthesis.